Okay, thanks. Yeah, so how many people are here more from like chemistry or origins of life? Okay. And how many people here from AI? Okay. Well, actually, that's more even than I thought. So this is, I put some stuff in to kind of introduce the methodology that we use in A-Life to study origin of life. But like, if there's something that is just completely, you know, what the heck is this, then like, please interrupt me, and I'll try to make sure that I explain it. So the stuff I'm going to talk about covers basically the last sort of 5 years of stuff I was involved in at ELSI, which is the Earth Life Science Institute in Tokyo. And it also involves this ELSI Origins Network, this EON project that was a kind of a subgroup at ELSI. So there are, you know, many, many people who I worked with directly or who, you know, we had discussions that contributed to these ideas. So I'd just like to acknowledge people who are involved in the work I'm going to present. So one of the big questions is we want to know in the origins of life, how did evolution arise? Because if we already have evolution and something sufficiently kind of, you know, enough moving parts to start, we could plausibly say that the rest of the understanding of the origin of life has to do with the processes of evolution operating on those parts and refining them and improving them. But if we actually need to get to that starting point where we didn't have evolution than we did, then that still needs to be explained through some other mechanism. It may be that it has something to do with like specific conditions. A lot of people say that there might be some special, you know, the early Earth looked in this kind of chemically perfect way and things happen spontaneously because of that, but on another planet they wouldn't. Or it might be something systematic about the way that the laws of physics and chemistry work and kind of organize the universe. So, you know, the big problem is if we have to jump all the way to something that looks like what a biologist would study with enzymes and DNA and RNA and proteins and cell membranes, that's already a lot. There's no way we're going to have that happen spontaneously without some kind of ordering explanation that goes above. It just happened to be that the initial conditions were right and then poof. So that's kind of one of the fundamental questions in thinking about the origin of life is like, how are we going to actually get to something that is simple enough that we can realistically have it, but has to actually exhibit the ability to get to what modern life eventually did. And there are a bunch of different ways to approach this problem. One approach is that you look at modern life and then you try to see like, okay, these things might have some common ancestor and their common ancestor might have some other common ancestor, and maybe I can like reconstruct backwards and break things down until I find the kind of the minimal organism that is in common with all life that we have today. And that gets you to something that's called LUCA, which is the last universal common ancestor, or sometimes people call it LUCAS, the last universal common ancestral set, which is a kind of realization that this might not be one specific, you know, bacterium that existed, but actually a population that was exchanging DNA and exchanging genes and so on. The problem is that actually is not old enough. We probably have at least 1 billion years of something happening before that, and that LUCA or Lucas is already extremely complex. So it's already got all of the modern synthesis type elements. It's got a ribosome, it's got cell membranes, it's got DNA, it's got RNA, proteins, enzymes, etc. So that it has this kind of, you know, in principle we should be able to go, or we would need to go further back than the data that is available in the modern world would let us go using this approach. Another approach is you could kind of try to make like a model early Earth and try to say, okay, what does the geology of the Earth give us in terms of chemical environments? And then can we get like specific chemistries that give rise to things in the lab? Can we make an amino acid from like CO2 atmosphere and lightning strikes and things like that? The problem is we kind of are making a choice of what to look for when we do that. What kind of chemical synthesis do we want to try to make work in early Earth? And that choice is based on modern life, not early life. So we might assume, okay, we should try to make RNA from early Earth conditions, but maybe there was some other molecule that eventually evolved into RNA. And then we would just totally miss it because we'd be looking for a chemical synthesis that directly jumps to this endpoint and just assumes that there was nothing in between. And a third approach, and I want to say none of these approaches are perfect, so I'm giving the downsides, but they're all going to have downsides. Although this third approach is the one that I take, so I'm going to be a bit biased here. is that we could actually try to generalize life. And we could say, like, if we could recreate the universe with our own laws, then, and we said that, yes, in fact, that universe did give rise to life, what would we need to have such that someone would be able to say, oh, because you got life, therefore your universe must have had this? And if we use computational methods, we can actually ask that kind of question. We can, take physics but turn off something, or take chemistry but turn off a reaction. And doing this, we can find, I should switch?
There is some problem with microphone.
Okay. All right, I'll switch to this. Hopefully this will be a bit more consistent. Yeah, so we can try to find the sort of minimal elements that we need to get life, or to get, maybe not life, but phenomena that are unique to life that we're aware of, like evolution and things like that. So this is the kind of the detail of, the sorts of approaches that people might use for that third method. The weakest thing we can do is we can just assume, we have compounds in a pot, they're mixed together, and we have one pot, so one set of environmental conditions and things like that, and reactions happen. This is one reaction. Imagine that you have many, many of those. Every time I have a reaction like that, I get a set of differential equations that basically say how the concentrations of the different compounds should change with time. If I have catalysis or something like that, I can do that at a network level by basically having A on both sides, or I could explicitly put some kind of mathematical model of catalysis on the reaction. And there are reasons why you would want to do both, or one or the other. And in this case, we've kind of gotten rid of a lot of the structure that you would assume in a biological system, like there's no spatial variation here. You can't be in one place and then be in another place and not interact. Everything is interacting all the time with everything else. There are no individuals. There's not even individual molecules because I'm not resolving that. There's just how much of each thing I have. There's no kind of self-assembly that's possible because I don't actually have the way to resolve what the energy of these molecules would be like next to each other as opposed to very far apart in that pot. Even though I do actually at least have catalysis. So this is like the bare bones. If I can get something in this, it almost requires nothing about the real universe. It's almost just this purely abstract mathematical model. But it has some properties we can identify, and even some things that look like properties of life. I can go a bit more, and I can actually say, well, maybe, the individual molecules matter. One thing that we will want to do, in general, is to have an open-ended chemical space. And if we do this kind of one-pot differential equation model, we have to identify what all the compounds are going to be ahead of time. But there might be some compound that occurs once in the entire universe because its length is, 200 or 300 carbons, and combinatorically, there's just no way to make that more than once. But if that molecule happens to be a catalyst or have some impact, then it matters if I have 1 or 0. So if I model things as individual molecules, then I can do this kind of bucket model where I pull sets of molecules out. I see if there's a plausible reaction involving those molecules. I calculate the probability of that reaction going or not going based on the rates and the energy involved in the reaction. And then I either update the system or not. There are more sophisticated ways of doing this. There's something called the Gillespie algorithm, which takes into account the fact that reaction rates in chemistry differ by many, many, many orders of magnitude. So if I just blindly sample molecules out of a bucket, most of them will have almost a 0 probability, and I'll just sit there waiting for the computer forever. But what I can do is I can sort of calculate what is the most likely next reaction to occur. And then I can calculate what is the distribution of times until that next reaction, and I can kind of do this event by event by event. So there are methods for handling even with very large rate contrasts. But when we go to, if we use this kind of thing, we still don't really have space. We still don't have self-assembly or any kind of macromolecular phenomena. Right? These things can't bend, for example. I couldn't have a pump if I wanted, or a membrane. So if I want to go there, people do molecular dynamics. That's another way you can go and try to capture these kind of more physical or spatial effects. The problem with this is that this is very expensive, and it's hard to actually predict anything by abstracting this because it's just everything is moving around. You might have some kind of general thing about the equilibrium of the system, but other than that, you just have to kind of simulate it and see. But you can get very, very kind of sophisticated things happening. And you don't have to always just do things based on the actual like force laws and coming from quantum mechanics on real molecules. You can write new sort of interaction rules and see what sorts of properties those very unphysical systems have in common with the physical universe. So like this is a artificial chemistry from Tim Hutton, and this is designed such that things move around like hard spheres, but when they collide, they might bond or they might not. And there's some set of rules for when things bond. And in this case, it's engineered. He was able to engineer something where you have some kind of information carrying molecule in the system. It gives rise to a membrane. The membrane encapsulates it, and the thing actually replicates such that the membrane replicates in sync with the information carrying molecule. But I should say, like, in this case, you shouldn't think this happens spontaneously. So the underlying this is a set of rules that is very specifically engineered to give rise to that exact behavior in the system. And if you change one rule, the entire thing breaks. So kind of another kind of approach that people use. And analyzing what a result like this means requires kind of thinking differently than if I was making like a physically realistic model. So that's sort of maybe a very, very brief summary of the kind of A-life approaches in artificial chemistry and trying to understand the origin of life. Now I'm going to go a bit more specific to the kinds of questions that I actually want to ask. Yeah?
Can you speak a little bit closer to the microphone?
Okay, like this? Okay. Sure.
So the title of the talk is Evolution Before Biology, but it's inspired by this paper, Evolution Before Genes. And this is an artificial chemistry like the ones that I described. So in this chemistry, you basically have ligation reactions and splitting, as well as catalysis and inhibition. And they have one other element, which is necessary for their results, which is that they put everything in vesicles, and the vesicles divide too. But there's no actual connection to the vesicles and the underlying chemistry, except that when there's a lot of stuff in a vesicle, it splits. So they don't say the vesicle is made of things produced by the chemistry. It's just this kind of background context that this chemistry lives in. There's a couple. So when they put these things in vesicles, what ends up happening is that the reaction volume is very small. So you have 1 or 2 or 3 of a molecule. You don't have 10 to the 8 or 10 to the 9 or 10 to the 20 of a molecule. And that means that things can completely disappear from the system or completely appear in the system. And they have this kind of sharpening effect due to these very, very tiny vesicles. What that ends up doing is it means that they actually get a sequence of kind of chemical sets that occur in the system. And they can show that the kinds of chemical sets that occur have something that looks a little bit like inheritance. So the set you get when a vesicle splits is somehow relatable back to the set that you had when you in the parent vesicle. And it also responds to selection pressures that they apply to the system. So they say that they can demonstrate an evolutionary process even though there's no explicit genetic carrier in the system. It's just concentrations of things or the presence or absence of different kinds of linear molecules. So no sequence-based heredity here, but still some kind of evidence of an evolutionary process. And they had to put in vesicles, and they had to put in catalysts and inhibitors. And I guess in the theme of the talk, and I'll come back to this point, what I would say is they've shown evolution before genes, but maybe we can even go further, because to get the kind of specific catalysis that they have, you would need something like an enzyme. You would need a large molecule physically. And inhibition is also really hard to get from a network perspective. You can get network catalysis fairly easily, but inhibition requires some very specific things if you want to get it through collective effects. So there you almost need the geometry of molecules and something blocking an active site or something like that. And maybe we can even go further back and not have vesicles, not have explicit catalysts or inhibitors or things like that, and still see the kind of evolution that they report. So I want to kind of investigate the possibility of going that direction and how we need to sort of think about or change how we think about the elements of evolution when we push it that far, so that we even lose the idea of individuality and populations and other kind of underlying things in biology. So that's kind of what to expect from the rest of the talk. So why do we want evolution? Why all the focus on evolution? Well, as I said before, if we can get there, there's a kind of idea, okay, maybe they'll carry us the rest of the way and we can just kind of rest on our laurels. But I think the bigger point is that we have 2 means of explanation of what happens in a system. One is that we start from some microscopics and do bottom up. So we say, here are the rules, here's the initial state. That initial state changes according to the rules, and when I iterate that many, many times, I get somewhere. And we basically can't really do the origin of life in a fully bottom-up way. It just requires too many coincidences if we're trying to get directly from rules and initial conditions to life. If I had maybe 1 billion years of lab time, maybe we could just look into it. But maybe it would only take a year. Maybe it would take 10,000 years. We don't know. With top-down explanations, we can kind of bridge large gaps. We can say, for instance, this function exists because it enables the system to dissipate energy. So when we talk about, for instance, if we were talking about like turbulent flow, and we look at how fluids organize themselves when they flow faster and faster and faster, when there's this kind of pressure potential to dissipate, what happens is that that potential energy becomes sufficient to give rise to vortices, and the vortices dissipate that energy more efficiently. I can also describe turbulent flow by starting from the Navier-Stokes equation and saying, well, this equation naturally gives rise to vortices under these conditions. But kind of both explanations have some degree that they let me get at something different about the phenomenon of turbulence. So that kind of top-down picture allows us to construct something like a statistical model of turbulence, and the bottom-up lets us construct like a detailed engineering simulation or things like that. They give us a different utility of navigating a difficult problem space. In biology, we might want to say that this function exists in an organism because it increases the organism's fitness. We say survival of the fittest is a principle that operates at least some of the time in biology, and we can kind of identify the boundaries where it's good and where it's bad. And so if we see that there's this function, we don't have to explain like exactly which genes changed and which bases to get there. We just say, okay, well, it's possible. It's within the search space of the organisms that existed at that point in time. And if it were discovered, it would be preserved. Therefore, seeing it has been explained, and we can move on. And we don't have to like try to reconstruct the exact location on Earth where that function was invented. So we can say, if we get evolution, that's kind of the earliest we can start using this top-down explanation. That's where we can start to engage this tool to cross a gap that exists in the origin of life problem. Okay, so as I was kind of leading into, genes are one biological element that we assumed we needed for evolution, but might not be so. And what other things are we taking for granted in this early evolution? What else can we get rid of? One thing is that in this chemical evolution picture, if I go all the way back, I certainly don't have genes or some kind of shared backbone that is where everything on that backbone is replicated in sync. But I also don't get individuals. I might say a molecule is an individual, but one molecule is not going to be a replicating organism. So that whatever collection of molecules working together to give rise to some kind of behavior or phenomenon, that thing, if it's an individual or if it's just like blurred together, that's something subject to the dynamics of the system. If I'm going to have individuals, it has to emerge. I don't have a clear kind of relationship between parent and offspring. If I have two things react, I don't have any kind of guarantee that the products of that reaction are like the things that reacted to give rise to it. In fact, they're going to be systematically different. They're going to be less reactive, a different part of the energy, certain kinds of potentials of changes are going to be kind of used up. So rather than having this kind of default of similarity, I've got this kind of default of a directionality of how things change as they move through this kind of reaction network, in the sense that the delta G is going to favor dropping in energy. But since I might have different numbers of reactants and products, it's not just like a simple everything goes downhill and I'm done, and especially out of equilibrium. And one of the big points of this is that selection, rather than being something that operates on lineages, like this lineage is good, this lineage is bad, this has more offspring, so it dominates at the end, I'm going to have something that's more like a tangled ecology. This thing might have some kind of local benefit, and as a result of the changes in concentration that come from that benefit, something else gets a benefit, and then that makes it suffer a penalty and so on. So I can't just say like this feature is good, this feature is bad. I need to actually think about the sort of matrix of interactions between things. And I actually have to map out what is actually subject to selection. Not everything in the system is actually going to be something that can be selected. And the second half of the talk is basically going to be going into detail and how we might measure that in a world without individuals. Okay, so the kind of core components that we do want to say something about at least. In Darwinian evolution, we have a source of variation, a mutation usually, but it can be other things. some of those variations are heritable. And because of those variations, a change in the rate of survival or propagation of the carrier of the variation happens. Right? So if I have a variation, but it causes a change in the survival rate of something else, that's not actually sufficient to make that thing go up or down in any kind of way that I can predict long term. It's actually the fact that it has that kind of closed loop, that the thing that carries it is also the thing that benefits or is hurt. And so we can take all of this mess in biology. It's not all of the mess. There's more mess. I only had so much space on the slide. So we have, the detailed molecular structure of cell membranes, DNA. This is the KEGG network, so it's all of the parts of the metabolic network we've mapped out. And all of that is just turned into how many kids do you have? And how many kids are your kids likely to have? And then when I do that, it turns out actually that's not so bad, as long as I have, individuals that I'm not in some kind of very complicated predator-prey ecological situation. I can say, okay, if on average, this lineage has 1.5 kids and that one has 1.2 kids, if I wait long enough, I'll have exponentially more of the 1.5 kid lineage than the 1.2. And that's basically the underlying structure. That's all I needed. That doesn't always work, even in biology. So multicellularity is kind of, if I map it out in kind of grotesque detail, I could say every cell in your body I'm going to track in the same kind of diagram, but then also at some point, you replicate too. And even if I take some region in there and I measure some kind of average fitness in a generation, it doesn't tell me anything at all about how the macroorganism is going to replicate, right? There is some kind of heterogeneity in the sequence, so that actually it's the contribution of that cell to the macroorganism that matters more in the long run rather than the cell's fitness itself. Having cancer doesn't mean you're more likely to have lots of grandchildren, even though the cancer cells themselves are, replicating more quickly than in someone without cancer. So that's one kind of failure of this abstraction. And horizontal gene transfer is another one that's actually quite all over the place in biology, where organisms will encode their genes into plasmids, release those genes into the environment, and other organisms can pick them up or incorporate them. It also happens through viruses, through sexual replication and crossover. So once you have that, this kind of simple picture of what's your expected number of offspring has to be modified in a certain way. And we can start to kind of ask, well, in these pictures, what's the unit? Are we talking about the evolution of a gene, the fitness of a gene? Are we talking about the evolution or fitness of a cell, of an organism, of an ecosystem? And that's going to be the kind of ambiguity that we will get in a chemical world. So in the biological story, the source of variation is implicitly weak. That is, the offspring are assumed to be, by default, kind of like their parents. There might be a mutation. Even high mutation is like, a couple hundred bases a generation would be a huge mutation rate probably in biology. But for a chemical system, it should basically be assumed that you almost have 100% mutation and you might have 1% similarity or something like that. And if you actually push that kind of mutation threshold very far in biology, this sort of survival of the fittest story actually breaks down. You get something called the eigenerror threshold, where even an advantageous mutation can't be retained by an evolving system. So if we're going to actually keep things around, whatever the mechanisms of heredity have to emerge from the chemistry, or we need forms of adaptation that don't need heredity to work, which I think is even maybe harder to imagine, but it's where there's a lot of interesting potential. We also kind of have this assumption that the heritable variations affect their carriers by default. If I make some new catalyst inside my cell, it affects my cell more than it affects some cell halfway across the world. But in these chemical mixtures, things are automatically interacting with each other all the time. Since we don't have individuals, when a new function emerges, it could be shared. It might have a bigger effect on the thing that gave rise to it because of what it does to everyone else, and because of what that effect does back on it, on the things that create it. So we have this kind of credit assignment problem, that I might invent some catalyst that would be really good for some protocell or some autocatalytic network or something like that. But if that catalyst can't create itself, it might as well just go away. It's not going to be, its function will not cause it to be amplified, even though it would be good in some sense to some kind of organism that we could propose might exist. So the traits have to actually be beneficial to themselves, not just to something, in order to be selected. And And even if we have something that's heritable and functional, the actual detailed network of how these interactions propagate may mean that there are just some things that can't be selected because of the dynamics. Okay, so it kind of gives us two classes of questions, which are going to be the two parts of the talk. One, how are discoveries preserved? That is to say, what do we have in place of sequence-based heredity? Is there any way to keep information alive in chemical systems? And if so, what does that look like? How does it behave? How does it scale? And also, what is the ability of a chemical system to discover things and to actually investigate changes in its own functionality and keep those changes if they're in some sense beneficial? What are the kind of the limits of the propagation of selection through a chemical system? All right, so thinking about heredity in these chemical systems. If I have some source, if I have some kind of, let's think of this as just like a temporal relationship or a causal relationship rather than a literal parent-child relationship. If I make some kind of change, I might have a system where that change immediately reverts because the thing that replicates actually is only replicating some aspects of it. So for instance, if I have a scar, my child won't have that scar, right? So that variation is not heritable. But on the other hand, because I have genes and genes are passed on, some mutation somewhere, a mutation in a germ cell basically can actually be carried by my children, by my children's children, and so on. And those are the things that can be sequentially explored by an evolutionary process. So that source of variation has to interact with a space that has the ability to preserve it. In this kind of chemical system, instead of having a sequence that copies itself with an explicit copy operation implemented by enzymes, I'm going to just have a system with dynamics. And in that kind of dynamical system, one type of object that exists or one type of feature of dynamical systems is they may have attractors. So an attractor basically means that if I put the system at some point in the state of, or the space of possible states that the system can be in, then it tends to go to a certain other point, and that many, many, many points distributed around that all converge to that same point. So if I have one attractor, it just means the system always goes to the same place. Not much you can do with that. But if I have two attractors, it means that in some sense, if I start near one, I stay near one. And if I start near another, I stay near another. And if I have something that is like spreading that system over space, like diffusion or vesicles that are splitting or something like that, then if my starting vesicle is near the first attractor, the vesicle that's produced may also stay near that first attractor. And in fact, that attractor may invade regions of the other attractor that are weaker in some way, that have a lower volume, or where the barrier to crossing attractors is less strong, or things like that. So one kind of model of a pre-biological form of heredity would be just, you say, how many attractors do you have? And if I have a mutation operator or something that can make me move between them, that's just the amount of heritable variation I can support in that system. I can even go weaker than this. I don't need these to be all equally good attractors. I just need to be able to say that when the environment is a certain way, one attractor is picked out, and if the environment or context were different, a different attractor would be picked out, but they might not have to be stable in each other's environment. So like, I could think if I have an organism that lives at very high pH or very low pH, those niches are kind of different from each other, and they might die in each other's niche, but kind of collectively, I can actually evolve to tune my biology to pH, because both are attractors that are potentially possible. They both have the ability to be stable in some kind of bigger context. So that's like kind of the weakest I can do anything with. But if I have attractors, it's even in some way gives me more than I need. So one model of this, one kind of prebiotic chemical model that has an attractor space like this is this GARD model. This is basically you've got a vesicle. Again, the vesicle keeps coming up, but it's made of different kinds of molecules. And each kind of molecule has some kind of preference for bringing others in. So each molecule catalyzes growth of the vesicle, but in ways that are compositionally different. So what can happen is you can get positive feedbacks. That is to say, if something, an obvious case is if like lipid type A brings more lipid type A in, and lipid type B brings more lipid type B in, then I'll get vesicles made entirely of A or entirely of B. But I can also have more complicated ones where A brings in B, B brings in C, C brings in A, or things like that. And as a result, these dynamics, if you simulate them, they can exhibit multiple attractors. So that's one kind of example that can give rise to this. So another example, more directly, is if I have a chemistry that's autocatalytic. So in this case, I actually have some compound that is directly producing more of itself from a food stock, maybe producing some waste. So a really simple example, if I have some A plus food goes to 2B, and then B goes to A in waste, that basically means I'm getting A goes to 2A. So A will exponentially grow. And if I have one of these, and I start in a world that has no A or B, just food or waste, I can't get A or B, basically. So I remember the fact that I had nothing. If I then put some A in, it grows, and it basically remembers the fact that there's A. So it has two attractors, although one of them is this kind of very unstable attractor. If, for instance, this were 2A plus food goes to 3B and something like that, I can actually have a region of stability of the zero attractor as well. So that's the difference between a first order and second order kind of autocatalysis. Can this give us heritable variation? So what we don't preserve with this kind of thing is variations in the details of the concentrations, only the presence or absence of the set. It's a strong exponential growth kind of effect, so it washes away little fluctuations in exactly how much A or how much B I have. If I change A relative to B, they'll basically return to some kind of steady state. And it turns out that if we make very, very big chemical networks, these things are actually quite common. We get lots of autocatalytic sets. But here's the problem. If I have one autocatalytic set, I kind of have one bit of heredity. It's present or absent. If I have two autocatalytic sets, I only have 2 bits of heredity if they have no way to actually ignite each other. So if the presence of A has some kind of byproduct that gives rise to some C that's also autocatalytic to itself, I can't independently turn A on and off and C on and off. If I turn A on, C won't necessarily be on. And if I make the network bigger and bigger and bigger, depending on the kind of statistics of the network, but for randomly connected networks, I pretty much most of the time will have this kind of cross reaction. So what ends up happening is that rather than the number of these bits of storage growing with the size of the chemistry, it grows slow. It basically grows like log of the number of compounds involved in the chemical network, at least for first order autocatalysis in kind of random networks. And the actual response curve says that if I, depending on how many reactions each molecule is involved in, I can have even worse than log n. So if I have too few or too many, if I have too few, I don't get any autocatalysis. If I have too many, everything becomes one giant autocatalytic set. And there's only like this narrow range, this critical point, where I actually get this log n scaling. So this is a pretty hard way to get much evolution in a prebiotic world. And this kind of consideration also may apply to something like GARD, where I'm basically looking to see whether or not those positive feedbacks are going to be isolated from each other or get kind of merged into one big blob. But at the same time, like, they didn't find this problem in the evolution before genes paper. So if I do a simulation of evolution before genes chemistry, I get all sorts of structure, all sorts of different attractors. I don't need that many compounds to get it. Why? Well, the results that I showed apply for first order autocatalytic networks, but the evolution before genes paper has something extra that I didn't consider in that model and isn't in the GARD model. It has inhibitors. And with inhibitors, rather than every time I add a compound, I add a new opportunity for 2 autocatalytic sets to become 1. By activating a compound, I may actually shut down some autocatalytic set. So that kind of breaks up the space. But again, inhibitors are kind of hard to get in a very primitive world because the actual network motifs for catalysts are quite common, but the network motifs for inhibitors require some very clean sort of local structures, or you need something that's happening at the level of like something blocking an active site. So you're already talking about kind of bigger molecules. So this is not a great answer. I mean, it still does get us further. It gets us to understand a little bit more what could give rise to these more these richer spaces where there's more evolutionary potential. But the other thing is, as I mentioned, potential attractors may be good enough. So if we only need one attractor to exist in any environment, a lot of these considerations go away. And that may mean that if we're looking for this kind of prebiotic heredity, rather than looking for heredity directly, we should actually be looking for this kind of diversity of attractors in response to different environments. Something where as we change the environment by a little bit, the kind of steady state of the system changes wildly and has many, many different directions of response. So a system that's extremely responsive rather than a system that has a really good memory. And that's kind of a different direction to look in, to look for these prebiotic evolutionary systems. Okay, so that's attractors and heredity and memory. But now let's think about selection and fitness. So in biology, we would say that the fitness formally is the expected number of offspring. And we can write down something called the price equation, which basically says if we have some trait and we know within a generation how that trait was correlated with the number of offspring, we can basically break down the change in the amount of that trait in the population between something that is due to selection effects, that is to say, I had more kids and the trait is unchanged, so there are more organisms in the next generation with that trait than before, and drift, which is traits change, and I basically say everything else is drift, everything where the traits change despite being in one lineage. I can essentially subtract that from that effect. So this is not a predictive equation as it is, it's just a post hoc thing. It explains that diagram in math. It says, given this many spheres of red color, blue color, green color, and this set of arrows that say how they relate between the generations, the number of red circles had to have changed by this much because that's just the sum of the effects. If, however, I actually had some way of saying that the fitness associated with this trait is always such, like I can predict that a future organism's fitness will be this because it has the trait, then this becomes an equation that predicts the future dynamics of the system. And that's where we might say survival of the fittest. This trait, gives a higher fitness, and that statement remains true. Therefore, we expect the thing with that trait to dominate at long times, right? So that's kind of the two sides of the price equation of this kind of description, that by having this concept of fitness and this ability to identify parent-child relationships in individuals, we can make predictions about what the system will look like at very, very long times, many generations down, as long as that kind of causal relationship that this trait causes this fitness remains true. This could also include the effects of something like me doing artificial selection. So I could use this just as well to say, I'm going to put an antibiotic on a plate, or I'm going to kill everything that is not expressing GFP, or something like that. So it's just sort of a description of the dynamics that I am enabled to do because I have these nice abstractions of children and parents and individuals. Of course, the pattern is I'm going to now remove all of that and see what I'm left with. If I want to do this kind of thing in chemistry, I have a problem. I have many problems. So I don't have any kind of clear association of traits into individuals. I don't have kind of unique parents. I also am drift dominated, basically. Most of the time, if I react a red and a blue, I won't get a red. I'll get something else. So basically, even though I could write down something like a price equation adjusted for this, most of the time, I'm not going to actually be able to have the things that let me use it to make good predictions. It's not actually giving me the power to predict anything anymore. It's just a statement about the dynamics. So this kind of suggests that what we need, rather than like trying to find literally the parent-child equivalents in chemistry, we just want to find something that gives us that same predictive power we had with the price equation. Or another way to put it is, what is fitness for? Why do we care? What should we use this term that we get to define to do? And again, this comes to this kind of top-down mode of explanation. We want to be able to say this trait exists because it would be good for its host. So if I have like some set of phenotypes and I know that they have differing fitness, I can just jump to, okay, highest fitness wins, that explains it. If I were doing this bottom up, I would have this detailed dynamics of exactly who begat whom, and I would still come to the same conclusion if my expectations about the fitness were true, but I would need to do so through a much more detailed resolution of the system that I don't necessarily have access to. And it works when traits cause fitness, not when fitness is just a post hoc observation of population count. So that causal element is needed for this to become predictive. And that's what we don't always have, where sometimes we get in trouble telling kind of fitness stories, both in biology and in chemistry. So, okay, we want to say, why is this compound present? It's like, why does this organism win? And we want to say that the question we're asking is, more specifically, are there cases where a compound is present because of its function? So in other words, did it kind of bring itself into stability or bring itself into existence or allow itself to remain through what it does rather than what it is? So an example of this would be if I have an autocatalytic thing and I remove that kind of autocatalytic network effect, it won't grow exponentially, I won't have it. But I could also have something where that's not as far as autocatalysis, like I have some kind of food stock, and all that's happening is that as that food stock has some kind of reaction pathways, there's a byproduct of the food stock of those reactions that accumulates. If that thing accelerates some reaction that is causing the food stock to actually be brought into the system, that also is going to have the same kind of feedback structure. On the other hand, if I have something else that's produced by that, and it has some other function that isn't part of this kind of feedback loop, and I take that function away, its concentration won't change at all. So I can kind of say, what if I intervene and see how the world changes as a consequence of my intervention, and use that to evaluate which things in the system are kind of giving rise to themselves, which things are there because of what they do, and which things are there just because they were put there by some other dynamics that could be assumed or that were just part of the context. Now, this is a tricky thing to ask because I'm saying, okay, let's go into the lab and change the laws of chemistry and do 2 comparative experiments. So how are we going to do that? On a computer, I can do it. In a model, I can do it. In actual chemistry, this kind of the question I want to leave up here is, how can we do it? Because I think putting it that way makes it sound really impossible, but at the same time, if we were talking about an enzyme, we could modify the shape of the active site without changing very much about the enzyme. So we could actually do this kind of intervention to turn off catalysis and see whether the enzyme went away. And the difficulty is that as we go to smaller and smaller molecules, I think we have to be more and more clever of how to do these kind of interventions in chemical systems. But I don't think it's impossible. So this is kind of what I want to ask you, since I'm a physicist and a theorist and an AI person, not an actual lab chemist. Are there ways that we can use this kind of thing to investigate the dynamics of selection in chemical systems? But I can do it in models, so at least I'll show you what happens, so you can have kind of an idea what motifs give rise to selective forces. So the basic protocol for a model is we have some kind of chemical network, some set of reactions. We initialize it and measure run it forward in time and measure concentrations. Then we take the same initial condition, but we change something about the catalytic relationships. We add a catalytic relationship, or we change a rate, or we remove a catalytic function. We measure the change in the concentration, and then we look to see when we added a function and the concentration went up, or removed a function and went down, we can say, kind of post hoc, that compound was there because of its function. That concentration was high because it did something, or that concentration was low because it did something. But if we get the kind of mixed effect, like add a function, the concentration goes down, or remove it, and it goes up, then we see sort of a selection against that. So the presence of the function causes the absence of the thing that makes that function exist in the system. We would expect to see fewer of those if we just ran a bunch of random chemistries, because that would basically say things that have that function should actually take themselves away from the system, so we should just not see them. And that's going to be the equivalent of the survival of the fittest in the chemical world. So, okay, so some very simple examples and then growing in complexity. This is about the simplest thing I can think to do. The boxes are buffers. So these are 3 compounds. One of them is buffered at a concentration of 1. That reacts to form this compound A. A reacts to form this other buffer, which is buffered at 0. So things are being injected into the system at the 1, removed from the system at the 0, and they transform through A. I have arrows here, but these are reversible, so it can go both. It can go both forward and backward. And what I'm going to do is I'm going to take every compound, which is A, and every reaction, which is the 1 goes to A and the A goes to 0. And I'm going to say, what if there was a catalytic relationship between that compound and that reaction? So what if A catalyzed the reaction that produces it? Well then, in fact, I get more A. Okay, easy selection for that particular function. If I had this kind of motif and there was the possibility of A catalyzing that, that would be selected for. What if I do it between A and the reaction that degrades A? Well, then, of course, it's selected against. This shouldn't be at all surprising. This should be the most trivial thing. So if this is surprising or unclear, I need to go and maybe explain something again. Okay? Okay. Then I can basically make more of these and see, okay, well, it doesn't really matter if I have many of them. It's many copies of the same thing. The inputs and outputs are buffered, so they're not in competition with each other at all. And in fact, if A catalyzes the production of B, it doesn't matter. There's no selection force there. Even though, in some sense, it would be convenient for B if it could somehow make A have that catalytic relationship. There's no mechanism in the actual dynamics of this chemistry for that to ever come about, so it just isn't selected. I can go a bit more complicated, and then it gets very, very messy. So I'm going to switch to these kind of color maps rather than showing the, rather than trying to do it through the dotted lines. Here I've got two autocatalytic chemistries. So A can form B, which forms C, which forms B with A again. So basically this thing here eats A to make more C and B. There's also one here where D and E eat A to make more of themselves. They'll exponentially grow up until the point where that decay process basically causes them to saturate out. And now if I can shut down B and C completely, that means it's not even competing with D and E anymore. So what I start to see is I can actually see things that are selected for killing other things in the system that are competing with them. And what I have in terms of the diagram here, I have on the y-axis, the source and the sink are the 1 and the 0, so the compounds, and then A, B, C, D, E, and then the reactions on the x-axis, and I just try every combination of what if this catalyzed that. So if A catalyzes source to A, it goes up, okay, easy. If B catalyzes source to A, it goes up even more, because the B, C, D, and E are kind of exponentially growing things, so their concentrations get very high, but A stays low. Those are pretty easy. Then you can see these kind of cross things, right? If D were to catalyze the formation of C and B from A, then it actually goes down, right? So if I help my enemy, I get worse, and there's a weak effect there. It's only about a 10% change in concentration as opposed to 40% or higher for these kind of direct inline catalysis. And similarly, if B can catalyze the degradation of E, right? So B can cause D and E to be driven out of the system. Because this is autocatalytic, it also reduces the rate at which D and E eat A. So in other words, it supports its own population and it goes up. So we can make this kind of map. If I change energies or reaction rates, then the details of the map change, right? But the kind of topological relationships, like what is good for what is bad for what, are pretty stable against changes in the details. So this is, to some extent at least, a topological feature of the chemical reaction network rather than a feature of the specific kinetics of the chemistry, or even the energetics. Although, again, the strength of selection pressures can vary a lot depending on where those things, how those energies hash out. So, now a very complicated example. Everything up to now didn't have dynamics, really. Everything, it's non-equilibrium, but it goes to a steady state. Oh, and I think it's worth mentioning that this kind of analysis, if the system is in equilibrium, all of the selective effect is 0 for everything, right? Because catalysis is just a change in rate. It's not a change in the equilibrium concentrations. So if I were in equilibrium, I would see nothing, no effect, no selection. So everything you're seeing here is pure non-equilibrium effect. Okay, so this is now a dynamical case. This is a chemical oscillator. It's a bit finicky to find this thing. Like, the topology is not hard, but I had to basically optimize the rate constants and the energies to actually find the oscillating regime. And what you can see is the dynamics of the 5 compounds for different scenarios, different catalytic scenarios. So of course the buffer at 0 is at 0. That's kind of boring. But then you have A, B, C, and D, and you can see it has these kind of wonky oscillations. But in many cases, the oscillations go. In many cases, the oscillations go very, very close to 0, and then recover after some time. So that kind of thing, if this is pushed down even a little bit more, it can just kill it. And in fact, you see some interesting kind of selection effects in this sort of system, because here you actually have the predator catalyzing its own degradation is selected for. Because if the predator is too effective, the prey spends more time in this valley where it's almost out, which means that the time average amount of prey available to the predator is lower. So if the predator can actually select against itself, it lowers the peak predator population, but it raises the average predator population. Right. Kind of an interesting effect. And this kind of model, although I'm presenting it as a chemical network, you would find something like this in something like the Lotka-Volterra equation, which is a sort of a biological or an ecological model. So it's the same kind of story. It's just instead of compounds, you've got rabbits and foxes or something like that. I think orange mites on the surface of an orange and flies is another example. All right, and this is all for small networks. And before I looked at how the heritability scaled in large networks to kind of see whether or not these are a way that we can get evolution in a prebiotic system. So it's natural to see what happens in large random networks for these selection effects. It's pretty sparse. Maybe not as bad as log n, but definitely most things are not selectable. There's just no positive or negative effect to have catalysis between that compound and that reaction. When you do see it, and these are two different random chemistries, when you do see it, you see this kind of vertical streak structure. And what that means is, if we were thinking about compounds as being the kind of equivalent of organisms, we might think that this compound has a property or that compound has a property. Selection operates on the compound. This is saying that actually it's better to think about selection operating on the reactions, as if the reactions were actually the carrier of heredity rather than the compounds. That's something which I think is going to take a bit more to cash out. And these are the stuff I showed in the first half were kind of old results, and these are actually fairly recent results. So what we're doing now with this is we're actually investigating how these patterns change when you, for instance, put these things into a vesicle, when you change the statistics of the chemical network, or if you even can evolve chemical networks to be like sort of optimally selectable. But it seems like there's a structure here that kind of goes against, at least for me, what I would tend to think would be the organizing structure in kind of a chemical story of evolution. It says that actually we should think about maybe ways that reactions could carry information, or the presence or absence of reactions in a system would carry information more so than the presence or absence of compounds. Okay, so in conclusion, there were two sections, one about heredity and how to think about heredity when we don't have a genetic system to work with. Attractor landscapes give us a way of doing that, but they don't scale very well. We escape that scaling in systems that have some kind of mechanism for specific inhibition, or if we think in terms of these potential attractors, and then we have to actually formulate, not just a dynamical systems theory in a given context, but the response of a dynamical system to the sort of manifold of possible contexts. And even if we do that, we also have to think very carefully about how credit assignment works, how selective forces propagate through chemical systems. To do this, we want to move, rather than a sort of a trait fitness type of model, into a causal model where we say, when does this thing cause itself, or when is the function that this thing has responsible for its presence in the system? As a result, we can kind of do this analysis, at least in simulated systems, and we find that the selective feedbacks are actually quite sparse. Many things that could carry function wouldn't be selected by a system if they did. And the functions have to be in very specific places, but those places seem to be concentrated more kind of systematically in reactions rather than individual compounds. And yeah, that's what I wanted to say, so thank you.
So thank you for interesting talk, and I think we have the time for discussion. So if someone has the question, I can give you the microphone because we are recording this. Nobody wants to ask? Ah, go on.
Nick, your large network is not that large.
Yeah, so I have to run one simulation for every point in the grid of every system. So it's basically n to the fourth to scale this up. I've taken it to, I think, maybe 150 or 100, something around there. Basically, the bigger it gets, the sparser it gets. So I don't have a scaling curve, which I'd really like to have. But that's something which, like, if it's, yeah, I think at the level of this, I mostly want to understand the motifs and things like this vertical striping pattern and how these things change. I think when it gets really big, it's going to be, you know, maybe even a different world. Sorry, my type of... Well, it's not that it becomes smaller and smaller, it's that the subnetwork that is actually subject to kind of evolution, these evolutionary forces is a smaller and smaller part of the network as I grow the network. So I get like more and more of the network is just kind of these leaf nodes that don't have feedbacks to themselves that actually cache out. And there are these subnetworks. And if I actually look at where these vertical stripes occur, it's actually kind of intuitive. They occur at bottlenecks. So if I've got like one big group of compounds and another big group of compounds, and there's one reaction that's responsible for like moving me from the one to the other, like imagine that I had, you know, nitrogen-containing compounds and non-nitrogen containing compounds, and in order to get from the one to the other, I need to do nitrogen fixation from N2, right? So then that reaction bottleneck that lets nitrogen enter the dynamics, that's going to end up being one of these vertical stripes. So whenever I find this kind of like organization into these kind of almost disconnected subregions of the chemistry, I get a vertical stripe. And for these kind of random networks, for the kind of the same, the autocatalytic sets don't scale very much. If I don't have like really, really severe, have emergent conservation laws, right? Like functional groups kind of act as a pseudo conservation law. It's harder to, you know, change the amount of NH4 I have in the system than it is to move it around. So if I don't have that kind of structure, then they get, you know, fewer and fewer and fewer as the chemistry gets larger. But if I actually had something where I saw a natural succession of, say, functional groups made of functional groups, et cetera, that were kind of pseudo-conserved, I would expect that kind of chemistry to have more of these vertical stripes, because it would have more bottlenecks or kind of important reactions that move you from one region of the chemical space to the other.
I think I have a lot more questions, but I'll leave it to other people.
Are there any other questions?
Hi, thanks for the talk. So I was wondering, how would you define a predator in your system? I was confused because I don't think I fully get what is actually flowing through your experimental system.
Okay. So in this case, it's by design rather than by observation. So if I have a complicated chemical network, I don't think it's clear to identify a predator. I think that's hard. You could look to see whether or not you have an anti-symmetric kind of selection effect. So like if this thing impacting that thing is blue, but that thing impacting this thing is red or something like that, that would suggest that like the benefit of one is the malice of the other. And that kind of anti-symmetric was in biology, if you have these kind of population genetics models, you might have an interaction matrix. And that interaction matrix, when it's anti-symmetric, looks more like something eating something else. And you get this kind of oscillatory response that you see in like Lotka-Volterra or like in this system. But here, this is designed to kind of be like a famous model in biology, where the predator is the one eating. And eating means that basically the reaction flows in a biased way from AB to the CD. Like if materials in AB, CD uptakes it. But if material's in CD, A can't uptake it. So in that sense, it's kind of by design.
Thank you. Maybe I misheard this, but does it also make to think about evolution before chemistry? You know,
evolution before chemistry?
Yes.
Yeah. Yeah. So there's an interesting. We were talking about this actually just before the talk. That there's this thing that Bob Hazen has found about mineral abundance, and minerals are kind of weirdly distributed in the sense that most minerals, by number, only occur one place on Earth. So they look like this incredibly diverse thing. They have the same kind of diversity distribution that biological organisms have. And in terms of this sort of thing that I was hinting at with potential attractors, we tried a computational system that was trying to explore this potential attractor idea. The idea is, it's still kind of chemical, but the idea is you have things that associate with each other. And then by associating with each other, they're somehow protected to stay in the system. But they have different kinds of patterns of association. So like, it's very much like GARD, except that now what's going to happen is that rather than like my presence brings in an arbitrarily large amount of this other thing, I have to pair with it. And so I actually get consumed in order to associate and protect myself in this reservoir. And what we found is that has this, it has no heredity, so it has one attractor, but it has this kind of rich potential attractor structure that I described. And it looks very much like elements precipitating out together to form minerals. It's kind of suggestive of that kind of dynamic. Like if you look at a packet of magmatic melt, and it's rising from the center of the Earth, or from the lower mantle. As that packet goes up, the pressure and temperature change, and things hit the phase diagram boundaries where it goes from liquid to solid. So when something solidifies, it pulls out some of an element that might actually have stayed liquid otherwise. And that changes the actual compositional profile in a way that as it goes even further up, the way that it successively crashes out might depend on very, very small variations in like the course that it took or things like that. And I kind of think that kind of pairwise association as a basic mechanism can give rise to some of these pseudo-evolutionary dynamics, and you wouldn't need chemistry for it, but you do need some sense of like things being things, which is maybe still a bit too chemical.
Are there any other questions, or if not?
Oh, no. Yeah.
I just wanted to ask if you turn it 2 slides forward to the reaction. Yeah.
This one?
Yeah, that one. It seems to me like the inhibitor relationship is there way more than the attractor one, like in the streaks especially. Is it just a property of these 2 particular models, or does it happen?
You mean
with
there's more red
model?
than blue?
Sorry?
Do you mean there's more red than blue, or?
Yeah, that it's more red than blue.
I'm actually having trouble reading that out, I guess, because the colors are quite, the dark red and the dark blue actually look kind of similar to me. Honestly, when I've looked at these things, red has been a bit rarer than blue. So these might be special examples. Red basically means that there's some kind of, like if I have something that can control its own input somehow, it's going to be blue, or it could be blue. But to have red, it means I have to have 2 things that could win that are different from each other. So like, I've seen a lot of things where I have like blue and a red pattern, and that blue-red pattern is mirrored on several reactions. So if I see like really well mixed red and blue, that actually suggests a richer chemistry to me. But yeah, it's hard to say, I think.
More questions? If not, let's thank Nicholas again for his talk.
Thank you.